Hello, and thanks for joining us today for the latest Technology Networks webinar, The Scars of Fat, the Transability of the 3D NASH Microtissues to Model Human Murine NASH and TGFbeta Signaling in a Jar. I'm Gareth Ginnigold, Senior Scientist responsible for our NASH projects at CM Bio, and I'm here to moderate the event. I'm pleased to have Michele Vacca to join us today as your presenter. Thanks for joining us, Michele. Dr. Vacca is a clinical research associate at the Institute of Metabolic Science at the University of Cambridge and an honorary consultant at the Cambridge University Hospitals. After his medical studies at the University of Bari, Michele specialized in internal medicine and completed his PhD in oncology and molecular and clinical pathology at the University of Chieti-Pescara. From 2008 to 2014, Michele was also Research Fellow of the Consorzio Mario Negri Sud and visiting clinician at the Ambulatory of Clinical Nutrition Clinica Medica Muri at the University of Bari. Michele's main clinical interest sits in the area of obesity and hepatic metabolism, particularly the interaction between obesity and the development of cardiometabolic and liver disease. His main scientific focus is the role of nuclear receptors, miRNAs, TGFbeta in hepatic and systemic metabolism and in liver disease. Following the webinar, we will have a Q&A session and we welcome any questions that you may have. You can submit questions for the Q&A session at any time during the presentation. To ask a question, you should enter your question into the box on the left-hand side of your screen and click Submit. We will answer as many as possible during the time available. For any questions we don't have time to get to, we will be sure you are contacted offline with an answer. If you experience any technical issues, please click the question mark at the top right-hand corner to request support. And please remember, you can ask questions at any time, even whilst watching the webinar on demand. So without further ado, I will now hand over to Michele.
Good morning, everyone, and thank you very much, Gareth, for the nice introduction. Today, I have been asked to provide my testimony regarding my collaboration with CM Bio that led to an external validation of their 3D NASH microtissues to model NASH and TGFbeta signaling in a jar. Before starting, please let me state that I have no commercial and financial conflicts of interest to declare, and that my relationship with CM Bio is purely based on scientific collaboration. My field of interest is represented by non-alcoholic fatty liver disease that has been recently renamed as metabolically associated fatty liver disease. We define fatty liver when more than 5% of hepatocytes carry lipid droplets, or if you prefer to look to the concentration of lipids inside the liver, when they exceed 55 milligrams per gram of tissue, which is the highest concentration that has been recorded in a lean, healthy individual. Non-alcoholic fatty liver disease is tightly associated with obesity and metabolic syndrome. It has been estimated that between 50 and 100% of subjects which are overweight obese with diabetes or with the combination of the two will also present NAFLD at the time of the diagnosis. Not only, if you look to the problem on the other way around, how many subjects with NASH are overweight obese with increased blood pressure or dyslipidemia, the numbers will not change between 50 and 100%. It's important to study NAFLD because it is associated to an increased mortality because of its intrinsic risk to increase cardiovascular disease, but also because up to 40% of subjects with the fatty liver will develop a pro-inflammatory and pro-fibrotic form of the disease known as non-alcoholic steatohepatitis, which has the potential to progress towards cirrhosis, and cirrhosis is a risk factor for hepatocellular carcinoma and for liver insufficiency, which are the two main causes why we request a liver transplant. Because of the obesity epidemic, over the years, we have observed a progressive increase of the number of admissions into the hospital because of NAFLD. And in US, also the number of transplants requested because of non-alcoholic steatohepatitis have been literally doubling over the last decade. If you consider that this dash bar is represented by hepatitis C, and the new antiviral treatments are preventing the progression toward advanced liver disease, so we are expecting this line to drop down. NAFLD is expected to become the first leading cause of transplants in the US and in the westernized world into the next. This is thus really important to understand where hepatic fat comes from and how NASH progresses towards cirrhosis. With regards to the sources of fat accumulation into the liver, we have recently published a review in gastroenterology where we claim that fat accumulation into the liver is all a matter of balance between influx and efflux. With regards to influx, it's really important the dysfunction of the adipose tissue that leads to peripheral adipose tissue insulin resistance, and peripherally, insulin is important to inhibit the hormone sensible lipase. When the adipose tissue becomes insulin resistant, a lot of free fatty acids are released into the bloodstream, and especially when we talk about the visceral adipose tissue, these will target the liver. Peripheral adipose tissue insulin resistance is also associated to hyperinsulinemia, and insulin into the liver will activate the sterol response element binding protein, which is an upstream regulator of de novo lipogenesis, the process that leads to fatty acid formation that then are used to build up complex lipids like triglycerides. Not only in a condition of hyperglycemia or in case of an enhanced dietary intake of refined carbohydrates, glucose and fructose are able to activate into the hepatocytes another transcription factor, the carbohydrate response element binding protein, that will turbo boost de novo lipogenesis. In terms of dietary intake, it's also important to look to the composition and the amount of fat that we eat. And also cholesterol is a main determinant, because it has been shown in animal models that if you eat more than 0.2 to 2% of cholesterol, this will lead to an enhanced production of oxysterols into the liver that will activate the liver X receptor, thus leading to the transcription of SRBP1, further boosting the de novo lipogenesis process. What happens into fatty liver is that associated to an enhanced influx, you will also observe a reduction of mitochondrial fatty acid oxidation, and also an impairment of the export of lipids into lipoproteins, such as VLDL. The Apothecides will try to counteract these processes, activating some less physiologic and less efficient metabolic programs, such as omega oxidation, which is important for very long fatty acids, and peroxisomal oxidation. These processes are not only less efficient in terms of production of ATP, but they are also associated to an enhanced production of reactive oxygen species that will lead to apoptosis, compensatory proliferation, senescence, and an enhanced production of pro-inflammatory mediators. These pro-inflammatory mediators will prime the hepatic resident macrophage that we call Kupffer cells, that will further amplify this pro-inflammatory wave, producing cytokines, chemokines, and growth factors that will lead to the recruitment from the bloodstream of different inflammatory cells. Kupffer cells will also produce a reasonable amount of TGFbeta ligands that will activate the hepatic stellate cells that usually are metabolic cells, but upon TGFbeta priming will transdifferentiate into myofibroblasts. Stellate cells will further amplify the pro-inflammatory milieu into the liver, and will also start to produce different collagen and extracellular matrix, thus leading to fibrosis and then cirrhosis. So there's no discussion that TGF-beta signaling is important for the progression of NASH. And it's not surprising that when we looked to the sequencing data of our bio-NASH cohort in Cambridge, and we clustered the patients against disease progression, mild NASH, NASH F1, F2, and advanced NASH, NASH F3 and 4, observed a stepwise increase into the activation of TGF-beta signaling and different downstream effectors of the pathway. But saying that TGF-beta signaling is activated in NASH is a really general concept. TGF-beta signaling pathway can be divided into two branches. We have the canonical SMAD2 and 3 pathway, which is led by TGF-beta and activins via ALK4 and 5, that is profibrotic, anti-inflammatory, and anti-proliferative. We have the so-called BMP pathway, for example, activated by BMP7 or BMP9, that is led via ALK3 and 6, that activate SMAD1, 5, and 9, that exerts completely counteracting effects, anti-fibrotic, pro-proliferative, and pro-inflammatory. Another thing to consider is that there are different ligands that can activate either branches of the TGF-beta pathway, and so far only few of them have been characterized into the pathophysiology of NASH. We got particularly interested into TVP lab at the IMS to one of the members of the family, and in specific the Bone Morphogenetic Protein 8B, which is completely absent into the normal liver. But looking to the patients against disease progression in two independent cohorts, we found that the level of expression of BMP that is almost absent into healthy fatty liver progressively increases with disease progression. Looking to its protein expression, BMP8B colocalizes with cytokeratin 18, hepatocytes, and alpha smooth muscle actin positive cells, thus meaning activated stellate cells. And the same kind of pattern of activation of expression can be also found into an animal model of NASH, namely the Western diet, which is characterized by refined carbohydrates, high saturated fat, and high cholesterol, 0.2. Looking to publicly available databases and clustering the data against the phenotype that has been described into the paper, it's pretty clear that the more is the hepatic damage, the more is BMP8B upregulation. Another important feature that attracted our attention is represented by the fact that in 2017, it was shown that both the BMP-A and BMP8B were able to activate both branches of the family. These evidences were produced in a different order. So first, we wanted to confirm this observation looking to the ligand binding, and we showed that BMP8B is able to bind to all type 1 and type 2 receptors, thus acting as a pan-agonist. And also, we confirmed into stellate cells, but not primary hepatocytes, that the BMP8B was able to induce the phosphorylation and the nuclear translocation of both SMAD2 and SMAD1, 5, and 9. So, since BMP8B is selectively upregulated in NASH, can potentially act as a rheostat of TGF-beta and the BMP signaling, and it's secreted and sensed by stellate cells, is BMP8B involved in NASH progression? The first evidence that we decided to generate was to try to understand which was the behavior of stellate cells in absence of BMP8B. Since we have the BMP knockout mice available into the lab, we decided to start this series of confirmation looking to primary isolated stellate cells from both wild-type literates and knockout. When you culture in vitro stellar cells into 2D, they spontaneously get activated around 8 days. And you can follow the process of stellar cell activation, scale of gray, looking to the expression of the alpha smooth muscle actin. We also confirm the observation at the protein level using immunofluorescence. Intriguingly, the BMP knockout mice, scale of blue, were characterized by a defective alpha-SMA expression both at RNA and protein level, and also a defective secretion of collagens like collagen 1 and collagen 3. Not only, we also recorded a defective expression of different pro-inflammatory cytokines like interleukin-6 and chemokines such as MCP1 and RANTES. A defective secretion of these cytokines and chemokines was also confirmed into the culturing media at similar time points. We then isolated the RNA from day 8 cultured stellar cells, and we performed the next generation sequencing. Looking to canonical pathways and upstream regulators, it was pretty clear that BMP8B absence not only impacts the activation of the TGFbeta signaling, this is not surprising, as you may imagine, we have knocked out a TGFbeta ligand, but also strong downregulation of different pro-inflammatory signals, such as TNF-alpha, interleukin-1 beta, interleukin-6 receptor, interleukin-8, and so on. As I explained you before, stellar cells are not only important for fibrosis, but also for the amplification of the vicious pro-inflammatory cycle. So we wanted to understand if the defect into the pro-inflammatory behavior of the stellar cells could have impacted also inflammatory cells independently and indirectly. To do so, we took the conditioning media from wild type and knockout cells at the different time points, and we challenged the in vitro activated macrophage that, as you can see in scale of gray, the more is the stellar cell activation, the more the profile of the macrophage becomes pro-inflammatory. However, into the knockout conditioned medium treated macrophage, it was pretty clear that this process was impacted, or if you prefer, delayed. So we provided evidence that the absence of BMP8B impacts the capability of stellar cells to promote a pro-inflammatory shift into the macrophage. Is this relevant also in vivo? We all know that these in vitro models have some limitations. To answer to this question, we decided to study the wound healing response following an acute liver insult with carbon tetrachloride. After the challenge, BMP8B was one of the most upregulated TGFbeta ligands. And the wound healing response is mainly characterized by inflammation, and we characterized a defect in inflammatory infiltrates into the knockout cells, into the knockout mice, mainly represented by monocytes and neutrophils. And also at gene expression analysis, again, a defect in different pro-inflammatory signals, but also into stellar cell activation. We then confirmed the defect at both 3 and 5 days in stellar cell activation, looking to the immunohistochemistry of alpha smooth muscle actin. And also we looked into compensatory liver regeneration. And at 5 days, we could recognize a defect into the expression of the proliferating cell nuclear antigen. We also confirmed the defect in liver regeneration in BMP knockout mice into the partial hepatectomy model, but I don't have time today to show you the results. You can find them into the publication. At this point, we wanted to understand if, given the effect of BMP8B on the wound healing response, and considering that the chronic activation of the wound healing response is crucial for NASH progression, if BMP8B could have been having a role there. Another important question was to understand how does BMP8B operate? And then the main issue to solve at that time was to understand how to study these paradigms in vitro in the context of NASH. Well, the main limitation was represented by the 2D cultures to deal with. And with this regard, the most tricky cell type to culture in vitro to get some meaningful data is represented by the hepatocytes. The reason for this is that the hepatocytes, when cultured in 2D, quickly differentiate. This is the levels of expression of albumin, and start to proliferate. It has been described that when you culture a tight confluence, these effects are minimized. However, in blue, you can clearly see that the process cannot be stopped. This has enormous implications when you try to study metabolism in a jar. And the reason for that is that the hepatocytes that become in 24 hours mainly hepatoblasts start to behave much more like a cancer cell, so a lot of anaerobic metabolism, they shut down the mitochondrial metabolism. And this is a pretty well-known limitation, at least from 40 years. And that's how I got in contact with CM Bio. A couple of years ago, they approached me asking me some independent evaluation of their preliminary data that now they have been published in Hepatology Communication. That's how the collaboration literally started. I found particularly intriguing the fact that he could culture in 3D scaffolds the hepatocytes, and also the fact that their MPS system was able to model the hepatic flow, adjusting it exactly to what you would expect in vivo. Another important thing that attracted my attention was represented by the high versatility of the scaffold that allowed to culture hepatocytes alone, or a combination of different cell types. And this was crucial for my experimental settings because BMP8B is mainly, like all the TGFbeta ligands, is mainly involved in a ping pong of signals among cell types. So I decided to outsource to them the experiment. CM Bio ran their standard NASH model with challenging the co-cultures of hepatocytes, Kupffer cells, and Stertula cells for 8 days with their proprietary culturing medium that is enriched in saturated and unsaturated fat, and also has physiological concentrations of insulin and glucose. And at day 8, they performed for me the challenges with BMP8B, plus or minus the inhibitors of both SMAD2 and 3 and SMAD1, 5, and 8 pathways. And then we also use some positive controls for both the signaling pathways to make sure that we would have been able to recapitulate the mechanisms that are associated to BMP action. We performed that treatment at 30 minutes to study phosphoproteomics, at 5 hours to study target genes of the SMAD, and 48 hours to study some long-term consequences. The first result that impressed me when I looked to the next generation sequencing data of the MPS system and I compared them with my human and murine data was the high overlap in terms of gene expression with the human and the murine NASH. This is particularly intriguing considering that in the MPS system we have only 3 cell types and more than 85% of the cells of the genes are overlapping among the different systems. Not only they beautifully develop lipid droplets, and looking to the top 100 genes, the most abundant transcript is represented by albumin. I have never seen an in vitro system that keeps such high differentiation of the hepatocytes. And you can clearly see that there is all the transcriptional machinery associated to the function that you would expect from a differentiated hepatocyte, like the production of apolipoproteins and their receptors, ATP transporters, coagulation and complement, different key metabolic enzymes, mitochondrial genes, and since this is a co-culture also with inflammatory genes and stellar cell genes, you also find different hits for these cell types, including alpha smooth muscle actin, TIMP, and different collagens. Moreover, really important for my experimental setting, the MPS system was also expressing all the the downstream machinery to TGFbeta signaling, and also different ligands of both BMPs and TGFbeta ligands, including the BMP8B that was my target of interest. Looking to the data after the challenge with the BMP8B, we were able to recapitulate the data that I observed into the stellar cells, also into the 3D co-culture. BMP8B is able to induce the phosphorylation of both SMAD1 and SMAD3, and at 5 hours, we can clearly see an upregulation, BMP8B is in orange, of ID1, a downstream target of SMAD1, and different downstream targets of SMAD3. Not only, but also BMP8B induces the phosphorylation of NFKB and STAT3, so it's not surprising that RANTES and MCP1 are promoted by BMP8B. And STAT3 is also a crucial upstream regulator of cell proliferation, for example, in liver regeneration. So it's not surprising that we also found an upregulation of c-Myc, cyclin A1, and different other genes involved in proliferation. The data regarding the pro-inflammatory action of BMP8B were also confirmed at 48 hours, looking to the secretion of different pro-inflammatory signals like the colony-stimulating factor, LCP1, and RANTES, while anti-inflammatory signals like interleukin-10 and interleukin-13 were suppressed upon BMP8B stimulation. Looking to the gene expression analysis and focusing on pathways and upstream regulators, it was pretty clear that on top of the stimulation of pro-inflammatory and TGFbeta signaling, different pathways involved in cell proliferation were activated. But if BMP8B promotes inflammation, is this relevant for NASH progression? To answer to this question, I moved to an in vivo model. Again, a Western diet model, 32 weeks, that models NASH F1. BMP8B, again, one of the most upregulated ligands in vivo, again produced by hepatocytes and stellar cells at RNA level. In terms of metabolism and liver weight, body weight, no differences between the knockout mice and the wild-type mice. We recognized that there was a reduction in liver damage as profiled by ALT, as confirmed histologically by the SAF score, specifically looking to the activity, which means inflammation plus ballooning, and also looking to the collagen accumulation with the Picos Sirius lab, we could detect a reduction of 30% of the total abundance of extracellular matrix inside the liver parenchyma. Looking to the liver infiltration, we also detected a reduction of the infiltration of B cells, T cells, and monocytes and neutrophils. And the defect of inflammatory signals was confirmed by qPCR, as well as the defect into collagen deposition. Summarizing all the data with the next generation sequencing and comparing the pathways which are activated into these wild-type mice by the challenges, carbon tetrachloride, partial hepatectomy, and Western diet, and comparing the knockout mice in the 3 challenges, it appeared really clear that the absence of BMP impacts inflammation, stellar cell activation and fibrosis, and cell proliferation in vivo. Intriguingly, we could recognize that the MPS system challenged with the recombinant BMP8B beautifully mirrored those findings, thus suggesting a high replicability of the results into in vivo models. Concluding, the human NASH microtissues cultured in the physiomimics model keep high degree of hepatocyte differentiation and functionality, beautifully mimic human NASH and animal models, and are suitable to study TGFbeta BMP pathways. NASH microtissues are cheaper and a faster option to study NASH pathophysiology, also in compliance with the 3 Rs. Together with CN Bio, we have shown that with system biology approaches and different layers of translation that BMP8B is a secreted peptide, so targetable, absent into the normal liver, so disease specific, and upregulated in hepatocytes and stellar cells in NASH, that acts as a rheostat of TGFbeta BMP signaling and coordinates the wound healing response, thus promoting inflammation and fibrosis in NASH, both in vivo and in the MPS system. I would like to finish acknowledging all the authors and collaborators of this story, and of course the founders. A big thank you to CN Bio that was crucial to provide me the framework of mechanistic confirmation that was requested by the editor to publish my story. Thank you very much for your attention. I would be more than happy to take your questions, if any.
Thank you very much, Michele. I'm sure you'll all agree that there were some excellent points raised during the presentation. We will now begin the Q&A session. To submit a question, you should enter your question into the box on the left-hand side of your screen and click Submit. We will answer as many questions as possible during the time available. We have had several questions submitted already, so I'll move straight on to these. So the first question is, can the CN Bio NASH model be used to understand key mechanistic data?
Hello. So I think that the answer is, at least for what I can speak about, which is TGFbeta and the BMP signaling, for sure, yes. And I was able to show into our story that it's pretty clear that SMAD phosphorylation occurs. At 5 hours, you can clearly see that the target genes of SMAD are upregulated, as you would expect. We were able to inhibit both the branches of the family using chemical inhibitors, thus affecting the transduction of the signal. So definitively for the TGF-beta signaling and for modeling NASH, the MPS system is a really nice model for preclinical science.
Excellent, thank you. The next question is the CN Bio system unique to allow the disease modeling of NASH?
For the models that I have so far tested, like cell lines, primary cells, both cultured in 2D or in 3D, I never found a system that was keeping, at least for the hepatocytes, such high level of differentiation. And also you can clearly see signals coming from the stellar cells and inflammatory cells. So at least for my experience so far, this is an excellent model to be the first step in any kind of preclinical research focusing on disease mechanism in NASH.
Excellent, thank you. So moving on, the next question is, would inhibiting BMP8B impair wound healing elsewhere?
That's a good point. We didn't produce any evidence so far looking to other organs, but absolutely this is a fascinating option. and, for example, thinking also to fibrosis occurring into the kidneys. Well, we know that it doesn't impact so much the fibrosis into the adipose tissue. But for kidneys where the level of expression is there, may be absolutely an option, also considering that it associates to obesity and chronic kidney disease, like, for example, secondary to diabetes. We didn't look specifically, but if you allow me to speculate, that's an intriguing development for the future.
Excellent, thank you. I'd just like to remind everybody that you can continue to submit your questions throughout the Q&A session by entering them into the box on the left-hand side of your screen. And now we'll move to the next question, which is, would you say this is a genuine alternative to animal models?
Well, that's a good point. What I can say is that if you look into the medium term, advanced 3D co-culture system will become the future of clinical science into liver disease. At the moment, we all know that if you want to publish a paper, you need to have also animal evidences, but this is looking into the medium term. In the long term, we are requested by the 3R principle to try to reduce as much as possible the number of animals and also to replace animal research. So absolutely, at least on the basis of our insights into this model, the data were with recombinant protein were beautifully mirroring the knockout mice. So this is a step that in the future, once enough data will be generated to further support our evidences, this is the trajectory that I think the field will take.
Excellent, thank you, Michele. The next question is, have you investigated patient hepatocytes for BMP8 upregulation?
So we investigated 2 independent cohorts, and in both the cohorts we saw in the whole liver. And in the whole liver, there was an upregulation. And as I showed in one of the first slides, hepatocytes were producing BMP8B as it was colocalizing both with albumin into the murine data and with cytokeratin 18 into the mice data, into the human data, but it was clearly hepatocytes looking to the phenotype of the cells. So yes, BMP8B is upregulated in human hepatocytes upon NASH progression. The more is the damage, the more is BMP8B upregulation, and this is confirmed in 2 independent cohorts.
Excellent, thank you. The next question we had is, the HSC activated by BMP8B, is it due to BMP8B secreted by hepatocytes or by HSC? Now, have you considered deleting BMP8B specifically in hepatocytes and seeing whether this reduces HSC activation and fibrosis?
That's absolutely a good point. So because of time constraints, I have shown also only the data associated to the possible autocrine effect of BMP8B. So primary stellar cells activated with or without coming from wild type and knockout cells. However, if you see into the extended figures of the publication, you will find also a challenge done with recombinant protein, both into the wild type and knockout cells. And into the knockout cells, you see a recovery, while in the wild type cells, you see a potentiation of the activation process. So we didn't use the strategy that was suggested by the colleague, but absolutely we have shown that if you top up with recombinant BMP8B coming from elsewhere, even the wild type cells have an acceleration of stellar cell activation.
Excellent, thank you. So I think we've got time for one more question, which is, could you comment on the reproducibility and reliability between culture batches using the CN Bio system?
I mean, we repeated the experiments twice, and both the times we ended up exactly with the same kind of results. This is the...
I can go into that one a bit more if you like. So generally we find with the same donor within an experiment, we get really good reproducibility. Obviously, one of the advantages of our system is that we get 12 wells on a single plate, and we can run 6 plates at the same time from one system, so we can run a lot of these things essentially at the same time, which means we get really good reproducibility. When you actually start using different donors, that's when you start to see some variability. Again, the things tend to trend the same, but that's kind of what you'd expect to see, is you kind of get donor variability as you would patient-to-patient variability. So as a general rule, yeah, the reliability is pretty good between different batches.
Okay, excellent. Thank you, Michaeli and Gareth. Unfortunately, that is all we have time for today, but I'd like to remind everybody that any questions we didn't have time to get to will be answered offline as soon as possible. And also, you can continue to ask questions even when watching the webinar on demand. The webinar will now be made available on demand. If you have any friends or colleagues that you think may be interested in this webinar or any future events, please feel free to share the link with them. I'd like to thank you all again for your time.